Okay, good morning, everybody, and welcome to my talk. I'm really pleased to be here today, and I'm really looking forward to this week, and which already started with exciting and interesting workshops on this past weekend. And I will be talking about taxonomy in the 21st century. So what is taxonomy? Taxonomy means giving names to organisms by associating existing names or by establishing new names for them. And by giving a name to an organism, we also link it automatically to a pre-existing classification, which has been provided by systematics. And in species level alpha taxonomy, the critical component for the correct linkage is the genus name as part of the binomial. And so the linear and binomial, I've argued actually in a recent paper, is a genius of combinatorics, but it's also half of the root of all evil. So if you look at the linear binomial, what is its power? So you have a combination of two words, and that poses actually the lowest effort on human memory capacity. If you imagine you have 1,000 species and you would have to name them with a single word each, you would have to memorize 1,000 words or number combinations if you want to do numbers. If you have two words, you can use as few as 63 individual words to name 1,000 species. The human brain can memorize on average about 50,000 words. So it means if taxonomists use 1% of their memory, 500 words for species names, that would be up to 500 species for uninomials and up to more than 60,000 for binomials, depending how the genus and species diversity is distributed. And also, while words have associations in the brain, numbers have none. So if you just think how many PIN numbers can you remember or memorize? Or which species has the registration number 394,901? I think it would do very difficult. We have a very difficult job if we had numbers as species names. What are the alternatives to the linear and binomial? Actually, there have been some papers, and this is a very nice one that summarizes some efforts and brings up some new ones from 1999, which with quite a lot of different alternatives. But if you look at these closely, 4 of these groups are not really different from the linear and binomial. They work exactly, they just have a different format. There are 2 more that are very likely confusing because they combine 2 different formats into one or use names and numbers. And there are also 2 that are really nonsensical. So really, if you think that these people that are actually top-level taxonomists have not been able to come up with a better alternative, we really have to accept that the linear binomial is the best we can have for alpha taxonomy. So what are the problems in modern taxonomy? Just to summarize, we have the genus name as part of binomial. We have, in some cases, lack of correlation between phenotype and characters in higher level clades. The typical hierarchical identification, we first go, for example, to family, like we do in plants, and the genus and species is often challenging, if not impossible in some cases. Species need to be, and so it leads to the fact that species needs to be identified before the genus is actually known. And there are specific toolkits required for accurate and precise identification, and these are often not accessible. So just 2 examples for this idea that species, you first identify the species, then you know the genus. In families Graphidaceae, for example, Graphis and Allographa have very similar species. They are not closely related, these 2 clades, and sometimes you really need to get to the species first before you know if it's Graphis or Allographa. And similar things happen in Pseudocysdaceae. As you all know, this is probably one of the most problematic families right now in terms of genus level classification, where you can have very similar things entirely unrelated. And also here, it's easier to get to the species, and then you basically look up what genus it is. And I just want to refer to Esther Geijer's talk on Tuesday, I think, about in the keynote speech about this family. So don't miss this. So now I want to go into some of the basics of taxonomy. First of all, accuracy and precision. What does this mean? Accuracy means I am in the correct clade. Precision means the level of resolution of the identification. So in this case, do you see this green lichen that looks like Lobaria pulmonaria? I would ID it as Lobaria. It would be inaccurate. It's the wrong clade, actually. And it would be imprecise because it's only to genus level. If I name it Lobaria pulmonaria, which I can tell you there are herbarium samples that have this name, it would be inaccurate. It would be precise to the species level, but of course it would be the wrong clade. Sticta would be the correct clade, so it would be accurate. It would be imprecise because it's only to genus level. And if I name it Sticta pseudolobaria, it's accurate and it's precise. So I think these definitions are very important when it comes to assessing the level of identification in a work. Another thing that is often confused is cryptic speciation and homoplasy. And I have here the example of Parmelia saxatilis morphodyme. Here you can see there's one clade as a monophyletic, and there's three species that look like saxatilis, and you can really call this cryptic speciation because it's from one monophyletic clade. But there's also other species that look like saxatilis in a different clade. And this would actually be homoplasy. It's not cryptic speciation. But in this case, we could also assume the whole thing is plasiomorphic. So it's really important that the context, the evolutionary context, is known before you talk about if something is cryptic speciation, if it's homoplasy or plasiomorphy, and that helps to assess the taxonomy of a group. Another important aspect when you look at trees is topology versus branch length. Topology only tells you one thing in the tree. So if you look at a cladogram, you see how things are related to each other. But if you look at the phylogram, the branch length, especially branch length, stem branches versus in-group branches, or within clade branches, is a very important feature to assess how you would translate phylogeny into a classification. And just to give you one example, here I have two exactly identical topologies, but with very different stem branch lengths. If A and B have the same morphology and C is a distinctive morphology, then grouping A and B together would be paraphyletic. Now, in this case, if the long stem branches, I would no longer call A and B paraphyletic because they both have obviously a long independent evolutionary history, and I would call these polyphyletic, not paraphyletic. Another thing is nested placement, which is often a problem at the genus and also at the species level. So let's assume A and C have the same morphology and B is phenotypically distinctive. We have a very different situation if B is quite closely nested within A and C, or B is on a very long stem branch. And this, of course, translates differently into a taxonomy. We can also have the situation that the placement is artifactual, depending on an analytical method. And there's actually one example that Manuela will show you in her talk today, actually. Shortly afternoon, about the photobionts in the Dictyonema subtribe. Another important aspect is splitting versus lumping. And again, we have the problem of paraphily in this case. So in this case, you have two clades A and B that are morphologically congruent and C is distinctive. And if you have a situation like this where C is nested within A and B, you can either split them into three or you can lump them into a single clade. And both actually have problems. So if you split them, then you have two clades, A and B, that are not diagnosable against each other. And if you lump them into one, which is often considered the better solution, we have the same problem, but the other way around. We have a heterogeneous group now where we have a distinctive element merged within a backbone of with a different phenotype. or morphology. So how can we assess this? One thing is a very simple approach based on information content. So in this case, for example, information content for the free taxa solution would be 1, and for the one taxa solution by default is 0, because you have only one higher group, a genus in this case. So why would we have information content of 1 on the left side? It's very simple. You have 3 possible comparisons, A with B, A with C, and B with C, and 2 of these are diagnostic, A and B against C, and 1 is not diagnostic, A and B, so it's 2 minus 1 is 1. In this case, splitting would be better than lumping. Now you can imagine a similar situation, but with more clades on the left side. In this case, the information content is 0, because you have 3 diagnostic and 3 non-diagnostic comparisons. It's 3 minus 3 is 0. In this case, splitting is equal to lumping, and you would probably go for lumping because it's the most conservative solution. Now, if you further increase your clades on the left side, you suddenly get negative information content, because here you have 4 diagnostic and 6 it's actually should say 6, not 3, non-diagnostic comparisons. So it's 4 minus 6 is minus 2. And in this case, lumping is better than splitting. So this is a very simple exercise you can use to decide which way to go. And one example is that the community is discussing about recently is it's a terariate core clade that has been recently lumped into just 2 genera from 15. And I will be talking about this in a short presentation on August 5th. Another interesting aspect is the enigma of methodological progress. If you look at the history of taxonomy, not only in fungi and lichens, but also other groups. So we started out, of course, with macromorphology, with the hand lens or the mounted lens, where we could see micromorphological features, compound microscope, chromatography, or say secondary chemistry, and then finally we have molecular methods and DNA sequencing. That's just, I think, the 5 major steps. If you look at these in context of different settings, you can say if you do field work, like most ecologists, naturalists, hobby taxonomists, you have the naked eye, and you may have a hand lens, and this may allow to see some micromorphological features, but not others. In a standard lab, which most taxonomists and maybe some ecologists have, you have microscopy. You may or may not have TLC set, but you usually don't have access to molecular methods. And then in an advanced lab, you have everything, and it's mostly evolutionary biologists. So one thing, if you look at this, is why are we treating DNA sequences differently from other methodological methods? So, and I just give you one example, which is the genus Pyranula, that has recently been revised. One group, the Ocraceoflava group, which are these orange Pyranulas that mostly occur in dry forests, by Ricardo Miranda Gonzalez and co-authors, which is a paper that is in preparation. And you see the 2 common species, Ocraceoflava and Ocraceoflavans, they differ only by ascospore features, ascospore size and septation, just a single character, and this has always been accepted. And then there's a third new species that has different ascospores and also has a different color, and this is caused by a different chemistry. So in flava and Ocraceoflava and Ocraceoflavans, we have perietine, and this new species we have chloramidine and amidine. So it's a second character you can add here. But even if they would be all the same in spores, you might even be able to accept just the chemistry as a differentiating character. Now, of course, if you look at DNA, you see that's really 3 different groups. This is a section of the mitochondrial SSU, small subunit. And so the question is, of course, these are all features. It's the ascospores, it's chemistry, it's DNA. So why are we treating DNA differently? And why do we not accept DNA as one important feature to perform taxonomy? And at this point, I just want to hint to another lecture, exactly by Ricardo Miranda Gonzalez, who has been also doing some interesting ecological studies with lichens in dry forests, and that should not be missed. It's on Friday afternoon. So what about if you think that most of these methods, the advanced methods, are not available to hobby taxonomists? Why should we care? And actually, there's a lot of reasons why we should care. First of all, hobby taxonomy is a multi-million-dollar business. If you just think about field guides, books, in how many copies these are sold, applications now on smartphones. We have citizen science, for example, as part of digitization efforts, the Herbonauts project, and for data collection. Especially, we can foster the connection to nature through taxonomy, and so people will be more attentive to conserving the environment, for example, iNaturalist and Mushroom Observer. And there's a lot of projects with environmental monitoring using citizen science, for example, the well-known Opal air survey in Great Britain. And so we really need to keep the folks hooked and not alienated with our taxonomy, which means we have to find solutions that work at different levels for different people and different users. And I just want to hint at the workshop that we had just this past weekend about iNaturalist. It was really nice and inspiring, and I think this is one of the great tools that are going to be used in the future quite often in taxonomy, and to link science with naturalists and take advantage of many different aspects of citizen science. So the question is, can accuracy and precision be compromised for the sake of practicability? And when do we need which level of accuracy and precision? And I just have listing here 5 different situations. We have human observations, for example, in these platforms like iNaturalist, Mushroom Observer. Very frequently, we have multi-species ecological studies. We have single species non-taxonomic studies in out ecology, ecophysiology, bioprospection, for example. We have taxonomic inventories and surveys, for example, mapping, environmental impact assessments, and we have alpha taxonomy. So that's the bread and butter of the taxonomic specialist. So I would say in single species studies and in alpha taxonomy, we need maximum level of precision and accuracy. In taxonomic inventories, we need a very high level. We can get away with a small number of species that may not be able to be identified. But usually, where possible, we get to the species level. And we should use the tools that are necessary for that. In multi-species ecological studies, it might usually be sufficient to have a high level of accuracy and precision. So there is a small amount of compromises we can make. And in human observations, due to the nature of this kind of data, which is mostly just photographs of the morphology, we have to accept in many cases a moderate level of accuracy and precision. So one important thing is, if a taxonomist is not actually doing the study, collaborate with a taxonomist, with an expert. It's a win-win situation. If you are not familiar with groups or are not a taxonomist, have no practice yourself, don't try and do this. And it's actually much better to involve taxonomists in, for example, in multi-species ecological studies, which not only improves the quality of the study, but also improves the impact taxonomic work has. And if we just take a step back and look at what we expect from taxonomy and what we can accept in taxonomy, and just putting here 3 situations. One is we have something in the field where we need the microscope or chemistry. We have to accept that we will not be able to identify that in the field unless we have a situation where the species are known in a locality and actually can be distinguished morphologically. In the tropics, for example, trying to do Agraphis or Allegrapha in the field is impossible. So this is okay. Now, if we have now a more frequent situation of cryptic speciation or homoplasy, for example, here in the familia succata complex, we will, in most cases, not be able to ascertain the species without DNA sequencing. And that is okay. If you want to get to the exact species, there's nothing else we can do. We need DNA sequencing. But what we don't want is this situation, where basically taxonomy expertise is completely lost and people only, basically taxonomy is automated and people think we can throw everything into a sequencer and see what comes out. So actually, this is not desirable. And so we have to ask ourselves, do we want a taxonomy that is ubiquitous? It can be done by everybody, or automatized. We just use a little machine like a tricoder. And at least in my opinion, I think this is not the desired goal. And I just give you one example when we try to employ molecular barcoding. So in this case, we used Usnia in Colombia, and we compared the result of just barcoding by blasting against sheet bank sequences, which is the first column. And then integrative taxonomy, where we combined actually multi-sequence alignment phylogeny, morphology, chemistry, anatomy, and type studies to arrive at the best possible identification. And when we set this to 100%, we see that the simple barcoding approach only gets 7% of this level. And the problem is mostly here, as we can see in what we call a barcoding wheel, is that most of these species have no match of the same species in the reference database. So what they come up with is the closest match, and the closest match is often far away, and that gives you, of course, then the wrong or uncertain identifications. So this idea of automatically identifying species with barcoding will only work once we have sequenced all species and we have accurate and complete databases. And that's a long way to go. That can only be completed by taxonomists and by experts. Now, if we ask ourselves, should do taxonomy, and I'm just comparing here with the situation of a medical diagnosis. If you feel sick and you want to know what you have, you don't go on the street and ask somebody on the street, or you don't ask your neighbor, you go to a doctor, you go to an expert, and you expect that this expert has the proper tools to diagnose your problem. So why should we treat taxonomy differently? So again, to summarize at this point, what are the problems that we're having? One main problem is phenotypically well-defined entities do not necessarily correlate with phylogeny. So this is mostly when we have genus level classifications that are where we cannot merge phylogeny and phenotype together in a single solution, but have to opt for either one or the other. And of course, we usually opt for phylogeny because we want to have natural entities as taxa. So in this case, we should apply integrative taxonomy to find the optimal balance between phenotype and phylogeny in terms of information content, not just senseless splitting, but also no senseless lumping. So the middle ground is always the best. And then we have the second case. It mostly applies to cryptic speciation, or where we cannot assess the characters necessary for identifying a species in the field. And so for accurate and precise taxonomy, we may require specific toolkits. And conceptually, there's no difference if this toolkit is a compound microscope, a TLC tank, or a DNA sequencer. And one thing we should say, accuracy is always a must. If something looks like something is a sticta, don't call it lobaria just because it looks like lobaria and you have no better idea. But for precision, we have to determine what level is needed in which situation, and then apply the toolkit required to achieve that precision. So if you want to get to species and DNA sequence is necessary to get to species, there's no alternative. And again, collaborate with taxonomists, experts, and this is a win-win situation for both sides. What does integrative taxonomy actually mean? So most people, or many people, think this means you do a phylogeny, then you classify, describe your species or discuss your species, and you put the discriminating characters in the discussion. Of course, it's important, but this is not what integrative taxonomy really means, because this is kind of an ad hoc discussion. What you actually should do is quantitative analysis of phenotype characters based on a molecular phylogenetic framework, which means you usually need a character table, and that is a lot of work, and this is the reason why many people don't do that. It's actually much more difficult to assemble such a character table than going to the lab and sequence a couple of 100 specimens. But there's a lot of integrative taxonomy toolkits that we can use. For example, of course, in molecular phylogeny, the combination of different markers, which should be functionally independent. So, for example, nuclear versus mitochondrial markers, ribosomal DNA versus protein coding markers. And of course, now we have, in many areas, we go into phylogenomics. What is also important is quantitative branch length analysis. And there's many tools that can be used, species delimitation methods, barcoding gap, temporal bending, all in combination, and all these are based on branch length patterns. And then, of course, as I said, we have phylogenomics, we have population genetics. For example, RedSec is now emerging as a very important method. And then, on top of this, we can, when we have the character matrix, of course, is a precondition for this. We can do character correlation analysis, look for redundancy, functional correlations, quantitative analysis of variation in phenotype characters based on a molecular phylogeny as framework. So plates as grouping variables, phylogenetic correlation, and other methods we can use. There's phenotype-based phylogenetic binning. There's other things like ancestral character state analysis. And then also, of course, we can distribution ecology as features. And there's quite a number of methods that can be used here. And this all can be done in the framework of integrative taxonomy. And I just want to give some examples and explain some of these approaches. So the phenotype-based phylogenetic binning is very interesting, which is a common situation. You have a group, a taxon, where you have a small number of species sequenced and a large number is not sequenced, but you have morphological or phenotype data for all of them. You can use several steps of an algorithm that actually first computes the morphological characters known for the ones where you have species where you have the sequences, and computes weights for these characters based on their distribution on the tree. And then it spins the remaining species based on these weighted characters onto this molecular tree. That gives a good idea, a good way to predict where species might end up in the tree, even if you don't have molecular data. And all of this works entirely in XML, so it's actually really easy to apply. Another thing is information content, where you want to do a more sophisticated approach than the one I showed before. Here's a situation where we have two genera or one genus Capsa in the family Delimited, which turned out to be polyphyletic. And here the question was, should we now distinguish two genera or 4 or up to 5 genera, depending on, or given the situation, how these clades are subdivided. So in this case, we use this MRPP, multiple response permutation procedure. And first of all, we compared the clade solution, 2 clades, 4 clades, and 5 clades, with maximum likelihood and maximum parsimony approaches. And then we looked at the number of discriminant characters that would apply. And you can see that in both cases, 4 or 5 clades is better than just 2. But between 4 and 5, there's really no difference. And so in this case, we opted for the 4 clade solution because it's the more conservative one, but it's clearly better than the 2 clade solution. Here's a case where we compared ITS barcoding versus phylogenomics. So this is two species, Sticta felix, Sticta lacera in New Zealand, that are morphologically quite distinct, but have more or less identical ITS. They cannot be resolved. So we use data from a 205 protein coding markers in the target capture approach. And we build pairwise comparisons within species and between species. And you can see that the Sticta lacera felix pair behaves more like a single species. So we try to explain why, if this is a single species, are there two discrete morphologies? And one idea was it might be an association with bryophyte mats. So if for example, the propagules germinate within bryophytes, the thallus is creeping along the substrate within the bryophytes and forms short branches, and if not, it actually becomes erect and forms a longer distinctive thallus that is perpendicular to the substrate, more or less. And so if we looked, when we looked at herbarium specimens, we actually could support this idea by a strong correlation between the morphology and the presence of bryophytes in the herbarium sample. But then a reviewer said, well, herbarium specimens are not really good to assess the ecological situation in the field, and suggested we should go to iNaturalist, which is what we did. And we had actually quite a number of samples to look at. And after confirming the individual identifications and the samples that were kept, we actually saw that there's not only no significant difference between the two morphs, but the felix morph is more frequently associated with bryophyte mats. So quite the opposite than what we found in herbarium samples, and not rarely the two grow close together. So there's really the argument that there's kind of an ecological modification went out of the window. But very important, the iNaturalist data contradicted the herbarium data. And so this is a nice example where you can incorporate citizen science into a scientific study. We still don't know what's going on here. So this is a case where actually the RADSEC approach might be interesting to use to look at this felix lacera problem. And this has been recently used very nicely in Pseudocephalaria species from the southern hemisphere, Pseudocephalaria glabra, where you can clearly see that this is a highly structured clade that might have actually more than one species, and this is a very highly resolving method. Has also been used in the case of Usnea antarctica and Usnea aurantiaco-atra. Now, another interesting approach you can do with an important approach with integrative taxonomy is, focusing on herbarium collections, historic collections, and also IUC assessment. And this is an example where we did both. So first of all, this is a species of Cora that is from Florida, and there's only historical collections, most of them going back to about 100 years or more, and some up to the 80s, and it has not been collected since then. And so Manuela was very successful in generating ITS sequences, both from Sanger and from high throughput sequencing from these historic samples, in part older than 100 years. We could actually see, confirm this is an undescribed species, also based on morphology. And based on the data we had, we could make rather detailed EUCN assessment. And we have an iNaturalist project actually asking people to looking for extant populations of the species. So this is a nice integration of many different methods. And obviously, it shows you that integrative taxonomy can tell a story. So this actually, if you try this, it increases the impact of your work, and you can get this into particular journals, and more people will read it, and not only those interested in taxonomy. And I just want to hint here at the symposium by Cecile Guedan and Manuela Dal Forno on tomorrow, I think, on this topic of herbarium collections. And Cecile will also give a keynote speech on this topic on Wednesday. And of course, we had the workshop over the weekend by Jessica Allen and Rebecca Yard. It was very interesting and very informative in how to perform IUCN assessments in red list species. And one very exciting aspect that is coming up now is machine learning. And oftentimes it's a bit difficult to envision what does machine learning actually mean. And it basically means that you have algorithms that instead of using predefined thresholds or conditions, they are trained through experience, through training samples, for example. And then you can use, for example, these methods to distinguish when you have like a phylogeny, you don't know exactly, is this like species or is intraspecific levels. You can use training samples to tell the algorithm to filter out what is species and what should better be recognized as infraspecies. And you can also use this, for example, at other levels to train an algorithm to tell you, what should be better recognized as a genus or a subgenus. So this is a very exciting development that I think will play a major role in taxonomy in the next 10 years or so. And here's also an interesting study that used machine learning to calculate the abundance of lichens in a tropical dry forest through near-infrared spectroscopy. My goodness. This is also a very interesting approach. I think there was no taxonomist involved here. I looked at the identifications and they seem to make sense, but this is of course something that where moderate accuracy and precisions are acceptable because it's more like of a proof of case study, but a collaboration with taxonomists in future studies would certainly be advantageous. And I think this is one of a promising approach to do large scale abundancy inventories. For example, if a set of species is known, it would be interesting to see how well the discrimination of this spectroscopy works actually at the species level. So if you look at taxonomy now and in the future, we can certainly say taxonomy is not a task that can be automated, at least not until all organisms on Earth have been fully cataloged and characterized, and we have complete and accurate reference databases for sequencing. And also when certain levels or proper levels of accuracy and precision are required, taxonomy should be performed by specialists. That is what they are here for. And that is why we need continued training of and support for new generations of taxonomists. And taxonomy should also be recognized as what it is. It's a complex and dynamic area of science, with continuous changes in methodology and also in the outcome. But it is converging onto a stable solution. And we should also think that taxonomy should, at some level, mediate between specialists and specialist solutions and laypersons, hobby taxonomists, naturalists, because these are the people who we can teach to care about nature and who determine our future. And to finalize, I want to give some practical recommendations, and I want to go back to this slide I have shown before in these different situations where we have different acceptable levels of accuracy and precision. And so how, if we have a lower level of accuracy and precision, how can we actually guide this? So first of all, for a specific situation, we have to define which level of accuracy and precision is required. Then we have to use the toolkits to achieve that level, which might be DNA. And we should always remember DNA is not black magic. It's just a tool similar to microscope or TLC tank. If toolkits are not available, or there are limited resources, or there's mismatch between effort and desired outcome, we can apply protocols for inaccurate and imprecise identifications, but they should follow certain standards. So for example, if a complex is unresolved, it has structure, but it's not classifiable. One example would be Cetraria aculeata. We can call this a species aggregate. If there's cryptic speciation, of a monophyletic clade, we can call this a species complex if we're not able to get to the actual species. If it's polyphyletic, we should call it a morphodeme, because we don't know if it's actually all in the same group. And if there's a character that cannot be assessed, for example, we only do field service, we cannot see ascosporochemistry, we might have to go to the level to genus only or species group. And all this is acceptable only when the context and the situation allow this. For example, if you do an ecological studies and different species in a complex have the same ecology, then it's fine to merge them together in one taxon or one group or one entity. But if they have different ecologies, of course, your approach will not work. And for the genus level taxonomy, we should seek optimal balance between phylogenetic structure and information content. So with that, I want to finish my talk, and I'm open to questions. For questions.